Hello, everybody. How's it going? My name's Alex. I'm joining you guys today from Ottawa in Canada, and I'm really excited that you could join us for a fun little class about cellular metabolism. I'm not alone here today teaching. I'm joined by two other awesome Kaplan instructors. We have Owen joining us from Missouri, and we have Carrie joining us from Texas. So if you guys have any questions during the lesson or anything like that, feel free to put your questions in the chat, and Owen and Carrie would be more than happy to answer them for you. All right, so we're gonna have a good time here, and without further ado, why don't we get started and talk a little bit about the MCAT and cellular metabolism. Cool, so today's agenda, talk a little bit about the MCAT in general. We're gonna dive in deep specifically into glycolysis and Krebs cycle, with a particular emphasis on the components that the MCAT likes to test about. All right, that's really what we're going to look about, because those two topics, I'm sure many of you have already, learned them many times, if not in some of your university classes or high school classes and so on. A lot of detail there. So we're really going to focus in on the types of information that are right for MCAT questions. And then we'll close off the lesson with some tips for success and discuss your studying in general. So without further ado, let's just really talk about why we're here. And that's because the MCAT is just a really, really important step in our academic future, right? This little pie chart here, nicely color-coded, shows the results of a survey of medical school admission officers that Kaplan has conducted, and you see that overwhelmingly, all right, they say that the MCAT is the most important factor. So that's why it's important that we take it very seriously. Cool, so what is tested on the MCAT? Well, we have 4 sections, right, of equal weight. One's chemical and physical foundations, basically general chemistry and physics, right? That's how we lead off the MCAT. We then go into critical analysis and reasoning skills. That's really just a fancy way of talking about reading comprehension, right? We got some biochemistry in this section, as well as the first one. We do see organic chemistry creep in. And then a section that is brand new since 2015, so about 5 years now, and that's psych and social behavior, right? So psychology, sociology, with a pretty strong emphasis on studies and experimental passages as well. Now, if you're looking at these number of questions, and you may have seen different stats with regards to the number of questions, that's because currently the MCAT is temporarily being administered under shortened versions, right, due to COVID-19 and all the administrative chaos that had ensued. But it is expected that the AMC will revert back to the full-length normal version of the test starting in January. So hopefully, fingers crossed, everything goes well. We could expect to see this, when you guys are ready to sit down for this big test, right? What you may notice is that all the sections are the same length and number of questions, with the small exception of the CARS section, right? And the reason for this is that CARS, reading comprehension, right? We can't have any questions without a text, right? So while all the science sections have freestanding questions, which here at Kaplan we like to call discrete questions, that's just not possible in CARS, and then that kind of explains the slightly shorter section. All right, how is it scored? Well, each section is scored from 118 to 132, right? It's a pretty random score. Now, we're looking at that, we're like, man, why such a weird range? Agreed, that creates weird numbers. But like most standardized tests, the MCAT has a normal distribution that is centered around a median score of 125, right? If you put all that together, right, the score from the 4 sections, that's going to give us a nice median score of 500. And that's why we have these weird looking ranges, because when you combine the scores of each section, right, we can easily, determine how far away we are from the median score, and hopefully in your case, above we are from 500. All right, so 500 is a solid benchmark, and that's why the combined scores are nice to compare against. All right, and of course, the reason it's saying 2015 there, it's not because this is the 2015, test, right? It's not like our slides are outdated by 5 years. That would be unfortunate. No, it's because 2015 was a year of major revision, right? Where the MCAT completely changed their test. In fact, it was the biggest overhaul, in MCAT history. So it was pretty serious. Right, what we have here on the board is a nice little breakdown of the relative weight of the various topics within each section, right? And what's interesting about this is you'll see that the topics are not really just segregated to their own sections, right? Like, for example, if we just look at biology, we see that biology, right, important information that we need to know from that general topic ends up being tested naturally in the biological and biochemical foundation section, but it also creeps up just a tiny bit. There's a small percentage in the chem and phys, and also a sizable, significant portion, right, of the psychology and sociology section, right? So that's why our big emphasis when we're learning towards MCAT success is not just going to be on mastering each section individually, but rather we need to be learning science in general, right? And learn to expect it in a bunch of different circumstances. Now, before we move on from this slide, I'll just want to give everyone just a little piece of advice with regards to how you're approaching your study, right? A lot of people like to, put focus on physics, for example, because physics, there's a lot of material that you need to know for the test, a lot of equations. A lot of people dread physics, but it's really important to keep things in perspective. Because if we look at physics, all right, it might be a huge, task to master it. Look at this bottom little bar. See that physics is a very small part of your overall score. It's really just a fraction of the MCAT, right? So this goes for everything. Keep in mind, right, while you're studying, to make sure that you're putting in lots of effort in sections that are going to give you the highest yield. In other words, the biggest bang for your buck, right? So definitely not saying to ignore physics. I'm just saying be careful in how much time you put into certain subjects. You want to make sure that you're focusing it on the subjects that are going to have the biggest payoff on testing. Right. So that's it for a little recap of the science content. And let's move forward. Oh, seems to have frozen. What happened there? Sorry about that, guys. It just seems like my slide has frozen for a second. There we go. Managed to switch it up. So MCAT is a lot more than just knowing science. All right. Honestly, if med schools wanted to test your ability to memorize science, they would simply look at your GPA, right? Assuming, of course, that you're coming from a science background, like most of you undoubtedly are. And if you're not, more than likely probably have to take a large number of science prerequisites, so they can check those anyways. Right, so a lot more than just knowing about science. It is important, right, which is why it's one of the cornerstones here, science knowledge. And if you were to go on the AMC website, you'd see that science knowledge is really one of the main skills, right? They call it skill 1, the AMC does. Skill 1 questions test your science knowledge. But on top of that, we have things called that the AMC likes to call skill 2 questions and skill 3 questions that look at how we're able to reason out, right, and problem solve using science as an accompanying partner, right? So we're going to be looking to solve problems in the world of science. So this little bar here, scientific reasoning and problem solving, shows a really important skill that the MCAT loves to test about. On top of that, many of the passages that the majority of the questions accompany, all right, are going to be experimental passages, right? So just to clarify, I didn't say that the majority of the passages are experimental ones. I was saying that on the MCAT, the majority of the questions accompany passages, and many of those passages are experimental, right? So that is, they're going to be describing particular experiments, some of them historical examples, some of them just randomly made up, right? And we're going to have to think about this and be like, oh, well, is this, legit? Do I like the way that this study was done? Has there been any, obvious flaws in the design that I can notice? Right? So just to give you an example of themes that might be talked about. Now, naturally, we're in election season in the United States. I say we, like I'm from Canada, not at all, right? So just to give you an example of a design flaw in a study that might happen is like, well, what happens if you want to do a poll, right, to see who everyone's voting for, and you don't account for the fact, that many people don't have landlines, right? So all you do is call people on their home landlines, right, without accounting for the fact that a lot of people are on cell phones. We would have a problem with sample bias, right? There would be a huge sample bias, probably ignoring, a lot of young people voting, right? So that type of reasoning and that type of awareness is something that the MCAT really likes to test to make sure that you're able to, think about a study critically, right? And like, why would they do this? Well, let's think about it. When you guys will be physicians, many years in the future, after having crushed the MCAT and crushed everything else that comes after it, right? You are going to get a huge volume of scientific papers and, studies to have to read. And you're going to have to, determine on your own, right? And I guess with your colleagues and see their opinion, if the studies that are coming up are legitimate, right? Like, are we going to use this, to treat our patients right away? Or do I think that there are some problems with this, right? And honestly, like, no better example of the importance of this, right, can come up than what we've seen in the last few months, with a multitude of new treatments for COVID-19 that are coming out very rapidly. Some of them, that haven't gone through the necessary rigors of approval, right? And that's okay. We're in a pandemic, so sometimes we need to rush these things, but it's up to the physicians to decide if they are comfortable with the way that these studies were done. So that's why the MCAT is making sure that we're comfortable with these type of ideas, right? And my favorite MCAT expertise, right, which is what's highlighted right now. Why is this my favorite? Because there's a misconception that the MCAT is super, super hard from a science standpoint only, right? Yes, you need to know your stuff, but just because a question looks really, really hard on the surface doesn't mean it's actually asking, all right, about the content that's there. And I'll show you guys an example of this once we get to our discussion on metabolism, right? There is a huge number of tricks, tips, patterns, language that comes up time and time and again in wrong answers, types of words that come over and over again in correct answers, right? And here at Kaplan, we're really huge on teaching those type of things to our students. So enough with the rambling by me about the MCAT. Why don't we get started on a little content review, right? We're going to be talking about glycolysis and the citric acid cycle, right? Also known as the Krebs cycle, also known as the tricarboxylic acid cycle, many names for it. So our primary learning goals for our lesson today are to identify key enzymes in glycolysis, right? Secondly, we're going to want to describe the steps and molecular regulation of the citric acid cycle. All right, so why do we say key enzymes? Why not every single enzyme? All right, and I'm sure many of you, all right, have memorized this perfectly, all right? I am sure, in fact, the majority of you have, right? But I use that in the past tense because I think we all know that once you memorize this, once you crush the test on university or in high school, it then fades towards the back of your memory, right? That's okay, right? Not everything is lost, right? Because once you learn something once, it's always a little bit easier to remember it in the future, right? Our emphasis here isn't to go on every single component of glycolysis. We're really going to focus on what the MCAT likes to test. And the MCAT likes to test the important enzymes, right? Important enzyme, I suppose, is a little bit of a subjective term. So when we say important, we mean enzymes that have significance in terms of regulating the flow through this cycle, right? Some key moments in glycolysis. And that's what we're going to look at. So without further ado, let's take a closer look at glycolysis. If we look at it from the molecular level, we see that while our overall goal here is to, or like our end goal with glycolysis is to produce energy, as well as to produce molecules that can feed other components of metabolism to create even more energy, right? That's our overall goal. We know that the molecule glucose contains lots of energy in its bonds, right? Breaking down a 6-carbon glucose molecule, right, in order to extract that energy is really the entire premise behind metabolism, right? Now, just because we're using glucose and extracting its energy doesn't mean, all right, we're not going to have to put energy in ourselves at some point, right? Glycolysis is almost like a, you know, those old email chains for the Nigerian like email scams with the princes. They're like, hey, like I have all this gold and I'd really like, I need to get out of the bank. And in order for it to work, you need to send me, you know, $2,000, right? I say back in the day, I'm pretty sure those still exist, right? Glycolysis is kind of like that, except it's not a scam. It's true. In order to get things started in glycolysis, we actually need to input a little bit of energy, right? And that's why you see here in our first step and a little bit later here, right, at the rate determining step, which we'll see in a moment, we actually need an input of ATP. But that's okay, because that's really just what it is, an input. We're going to get far more out of it than we put in at the start. So that's something to keep in mind. ATP goes in, but ATP also comes out, right? And we get more out than what we put in. All right, so let's get started on the first part, which is what we like to call, you know, like the preparatory phase, all right? We're gonna prepare the situation for us to be able to extract all this wonderful energy, all right? That's our point right now. So step one, we have glucose, right? Glucose, get this from our food. All right, so you eat it, like say something delicious full of glucose, I don't know, some fruit or something gets broken down, or some milk, you know, it's got some glucose in it, or anyways, the glucose is part of the components that make up milk. All right, so we have glucose. First thing we need to do is we need to phosphorylate the glucose molecule. We use ATP for this because ATP is what's going to be providing that phosphate, right? Fortunately, glucose doesn't just get phosphorylated on its own, right? We need the help of a special enzyme called hexokinase, right? So one thing I'm really big on, instead of just shoving, you know, enzymes in our brain, try and remember one cool thing about that enzyme, right? And I use the term cool very, very liberally here. When I like just a fun fact about an enzyme or that step, right, that can help you put things in context. Much easier to remember things that way, right? So it's up to you to decide what your fun fact is going to be. But for example, I think that what's really cool about hexokinase is that it has the prefix hexo, right? Everyone just take a moment to think, what other thing do they know of in their, like, that they've learned about that starts with the word hexo? Cool. There's no perfect answer to the question, but when I think hexo, I think hexagon, right? A 6-sided polygon, right? And hexokinase is called that because it phosphorylates a 6-carbon molecule And the word hexo is further there because it adds that phosphate on the carbon in the number 6 position. So that's why we use this enzyme, and that's why it's called hexokinase. Cool. Furthermore, we see this weird little Mg2+, right? Many of the enzymes that are involved in cellular metabolism, right, require divalent metal cofactors, right? In this case, hexokinase uses magnesium, right? The exact specifics of why this is important are beyond the scope of the MCAT. Let's just say, for our overall understanding, that phosphate has many oxygens on it, right? Phosphate has lots of oxygens, and these oxygens carry lots of electrons and negative charge, right? All of this negative electron clouds, right, really, cramps the style of phosphate and its interactions with other molecules. So the role of these positive cations that are associated with these enzymes is to essentially protect the phosphate, right, from all the negative charge that's cramping its style, right? They're basically protectors. Cool. So that's how we get started. ATP is hydrolyzed, right? We're breaking it. ATP goes, this is adenosine triphosphate. So it's a molecule that has 3 phosphates on it. And it becomes adenosine diphosphate because we are poaching one of those phosphates and attaching it firmly onto the glucose in the 6th position. All right. Why are we doing this? All right. It's not just important to memorize the step, but trying to get context as to why this step is important is the key to long-term retention, but also the key to be able to tackle use the content to tackle MCAT style questions, right? Well, the reason we do this, right, is to essentially trap the glucose in the cell, right? How does this trap it? Well, remember, glucose enters the cell, right, through the action of transport proteins, right? We call them GLUTs, right? There's different types of transporters, right? But glucose transporters, they move glucose. They are fairly specific to the point where while they can move glucose, they cannot move glucose 6-phosphate. And that's why this step happens. It traps glucose in the cell and say, all right, well, you got nowhere else to go, buddy, right? Now you're going to have to be glycolyzed, right? To use, it's not a real term, but yeah, that's what's going on. Cool, right? And one more thing that's important to note is that hexokinase is part of a process that is irreversible, right? Irreversible. I put so much emphasis on this because the MCAT likes to ask us about which processes are reversible and which aren't. Now, instead of learning, the specifics of every single step, I find it a lot easier to just remember that in glycolysis, we have 3 irreversible processes. This is one of them. Everything else can go forwards or backwards. All right, cool. So lastly, before we move on, it's important to talk about regulation, right? Typical MCAT questions, right? Like not every question, of course, but a typical theme that the MCAT likes to ask about is relationships, all right? What effect would, increasing glucose 6-phosphate have? Would that cause an increase in activity in hexokinase or a decrease in activity? Like those type of questions. They love the increase, decrease based on a new situation. So glucose 6-phosphate here has negative impact on the action of hexokinase. And that makes sense, right? We don't want to start making more and more glucose 6-phosphate, right, and get a buildup of stuff, right, when we're not ready to use it, right? And this goes true for cellular metabolism. This is true for other processes in the body, and also many real life situations, right? One of my friends was telling me that, last year when there was a lot of flooding in the Ottawa, like New York region, like around the Great Lakes and the St. Lawrence region, a lot of flooding, and a lot of government entities, that would live downstream would be, yelling at the other government entities living upstream, being like, open the dams, or close the dams. We don't want more water coming, close your dams. And they're like, we're opening our dams because like we have too much water, right? So, the government's yelling at, other entities that are living upstream would be in the same kind of situation that's going on here with glucose 6-phosphate. Be like, stop sending us water. In this case, stop sending us more glucose 6-phosphate. We've got our hands full. Right. So that's first step here. Glucose 6-phosphate gets turned into fructose 6-phosphate by an isomerase, right? We're not going to focus on that because it's really not too much interesting there, reversible process. We're going to draw our attention here on this next step, the conversion of fructose 6-phosphate to fructose 1,6-biphosphate, right? Why are we focusing on this? Because this is the key step in glycolysis, right? There's never been a more important step in glycolysis than this one, right? So it's step 3 because we just quickly, glossed over step 2, which was turning our glucose molecule, glucose 6-phosphate, into fructose 6-phosphate. Right, so fructose 6-phosphate, we turn this into fructose 1,6-biphosphate. Right, so everyone just take a look at what we have here on the left and on the right, and just based on what it looks like, try and figure out exactly what has happened. All right, so if you might notice, we have an extra number here. You see we've got a 1,6 and then a 6. Why is that? We've added an extra phosphate. Okay, so we've added an extra phosphate. Where did that phosphate come from? It came from ATP again. Okay, so ATP, all right, gets hydrolyzed, and we've phosphorylated our fructose molecule. Who has helped us do this? Well, it's our friend phosphofructokinase 1, all right? For short, we call it PFK1. This is the master enzyme in glycolysis, right? Why do we call it the master enzyme? Because it is the rate determining step, right? Like once this gets going, well, we're going. Right. Once again, we still have the cofactor of magnesium, right, to help shield the phosphate from the very dense, the very negative electron clouds surrounding all those oxygens. Right, so that's clutch. So yeah, ATP is hydrolyzed. Once again, this is an irreversible reaction, right? It is, though not always the case, it is very common for when you need to burn energy, right, by like, using up an ATP molecule. Right, it makes it very difficult to go in the reverse direction when that is occurring. Cool. It's kind of think about it this way: it's not too bad. Like you could burn a piece of wood, right, without too much trouble, extract all that energy. It's kind of difficult to put all the energy and the embers and the ash together, and the carbon dioxide that's gone off, put it all together and go backwards. Right, similar situation here. Right? And as I mentioned before, this is the rate limiting step in glycolysis. Cool. All right. So really, really important. Phosphofructokinase is probably the most important enzyme to remember. And this is the second of the third irreversible steps in glycolysis. We saw hexokinase just before, and now we're seeing phosphofructokinase 1. All right. Talking about the regulation here, it's actually pretty interesting. So we can have positive stimulation of this conversion by AMP, right? AMP adenosine monophosphate, basically just ADP with one phosphate less. All right. Everyone take a moment to try and think about why that is the case. All right, well, we need to think, well, why would we want to go through this process? Right, we'd say, well, we want to go through this process when we're low on energy, right? And AMP is missing phosphates, right? ATP is how energy is stored in the body. That's like our energy stores, essentially, in the short, not our long-term stores, but like ATP is how we deliver energy to the places that need it in the body, right? So if we don't have a lot of it, it means we need to make more. So it stands to reason, right, that when you have a lot of AMP, which is functionally depleted ATP, right, you would want to fill it back up. And that's why high levels of AMP, all right, have a positive effect on the rate limiting step in glycolysis, right? By the same token, right, if we have lots of ATP molecules, right, plenty to go around, stands to reason as well that this would have an inhibitory effect on phosphofructokinase. Cool. Something that's kind of interesting, though not depicted on the slide, is that a component of the Krebs cycle, citrate, also has an inhibitory effect on phosphofructokinase. Right? We don't see it on the slide here. It's not a big deal. All right, cool. Now, what's going on here? We see the presence of another little enzyme, phosphofructokinase 2, in the liver. Okay, well, why is this? This is pretty interesting, actually. I find this kind of cool. We talked about the inhibitory effect of ATP and also citrate on phosphofructokinase. When we have lots of energy, the body doesn't want to be doing things for no reason. It's like, hey, stop, we're good, right? But the liver is saying like, okay, cool, I know we're good on energy here, right? But like, I still use intermediates from glycolysis to make a lot of other cool stuff, right? I use it to make glycogen, I use it to make glycerol, I use it to make fatty acids and all that cool stuff, right? So while we may be good on energy, right? Like I could use those things to store it for later, right? But you've just shut down the whole operation. So what happens is that this little enzyme that was only found in the liver, phosphofructokinase 2, right? It actually takes just a tiny bit of our fructose 1,6-biphosphate and turns it into fructose 2,6-biphosphate, right? And the whole point of this is that this here can then say, well, you know what? I know you were saying to stop make it, but we're changing the plan, right? So in essence here, this is a bypass to all the inhibitory effects that too much ATP and products of glycolysis would generally, create. So it's essentially a bypass because the liver needs to be making some other things, right? And the regulation of this is controlled by other hormones such as insulin and so on, but minor details, right? Focus on the main ideas, right? I cannot stress enough. These are not things you just want to memorize one after another. You're going to go nuts, right? Learn the big picture, right? And plan to look at these type of pathways multiple times, right? We want to look at these pathways 2, 3, 4, 10 times, right? Adding little pieces every time we look at it. Trying to learn it all at once, you're just going to be miserable, right? And I cannot stress this enough. All right, cool. So we talked about phosphofructokinase. It's irreversible, right? Rate limiting step. Creates fructose 1,6-biphosphate, right? And then this molecule, which is still 6 carbons, right? We still have a 6-carbon molecule, is going to get broken down into a 3-carbon molecule called glyceraldehyde 3-phosphate, and another molecule called dihydroxyacetone phosphate, or DHAP for short. Right, now, when we're dealing with DHAP, we have 2 options, right? Option 1, right? DHAP it's used for other pathways. Here we just say alternative pathways, right? If you'd like to learn them, you can do that another time, right? Just in essence, it could be used to make glycerol, right? And then triglyceride synthesis and so on, right? If you don't want to do that, well, then there is another enzyme called glycerol 3P dehydrogenase. Don't worry about the name, not a really important one. Can then convert this DHAP into some glyceraldehyde 3-phosphate, right? What does this mean, big picture? Well, big picture, what this means that we have gone from 1 glucose molecule at the start, and we've now broke it down to a point where we have 2 3-carbon molecules called glyceraldehyde 3-phosphate. So we're down to 2 3-carbon molecules. So remember now, for every 1 glucose that comes in, we now have 2 of these. So everything that comes up from now on in our payoff step is going to be happening in double for every 1 glucose that went in. Something to remember. So that's why we have a little ×2 here on the slide. All right, cool. This brings us to our payoff phase. All right, what does payoff mean? Payoff means that this is where we're going to get all the good stuff. We had to put in a little effort first. We had to put in a little ATP, right? But now we're going to reap the rewards from all this fantastic, setup that we've done. So let's get in on it. All right, this is the payoff phase, right? And we're going to start with the conversion of glyceraldehyde 3-phosphate to 1,3-biphosphoglycerate. So we're on to step 6. Cool. So what's going on here? We have glyceride 3-phosphate, all right? And we are going to be making an NADH molecule using the enzyme glyceraldehyde 3-phosphate dehydrogenase. Now, little pro tip, all right? Notice, just before I get to the pro tip, everyone take a moment to look at this molecule here, NAD plus and NADH. Everyone take a moment to look at that. Take a moment to think about what the difference is between those two molecules. Everyone see that the difference between the NAD plus and the NADH is a hydrogen, right? Where did that hydrogen come from? We poached it from the glyceraldehyde by oxidizing it, right? So we oxidize the glyceraldehyde and we're gonna reduce the NAD plus, right? Where does the pro tip come in? Well, in order to take a hydrogen from this, we need to use an enzyme that's gonna dehydrogenate it. So almost always in these metabolic pathways, when you create an NADH molecule, right, it's being done through a dehydrogenase molecule. Sorry, a dehydrogenase enzyme. And this is clutch because there's almost like the main point of cellular metabolism is to create high energy electron carriers, NADH and FADH2, all right, which are then used to create a proton gradient in oxidative phosphorylation, electron transport, and ultimately ATP energy, right? So there are a lot of enzymes to memorize, but you'll notice that many of them have names that help us deduce their function, right? And help us, anticipate, I guess, what they would be called. And anticipating the function of an enzyme based on its name or its role is going to help us answer MCAT questions without necessarily needing absolute total mastery of the content like you may need in a college course about similar material. So we have a dehydrogenase. Cool. All right, so NADH is being generated. That's clear. And this NADH will then be used in electron transport chain, right, to help create a proton gradient, right? We're producing a very, very high energy intermediate, 1,3-biphosphoglycerate, right? And this is reversible. It's not one of the 3 reversible steps in glycolysis. Okay, a couple things before we move on here. I want everyone to look again at this glyceraldehyde 3-phosphate, the 2 rectangles in green, and 1,3-biphosphoglycerate, and see what is the main difference, right? Something we've seen before. Everyone see that there is an extra number here, the 1, which means that we must have phosphorylated this somehow, right? So there's actually something else that's not in the slide, right? We do have the need for an input of an inorganic phosphate in order to phosphorylate this molecule. Now, this actually requires some energy, but we see we don't even have energy. We don't have like ATP or anything on the slide, right? Furthermore, we have the reverse of energy. We're actually storing energy in a high energy electron carrier, NADH. So where's all this energy coming from for the phosphorylation to create a high energy intermediate in 1,3-biphosphoglycerate and, reducing an NADH molecule? Where's this energy coming from? It's coming from the oxidation of glyceraldehyde, right? So this is basically what's happening throughout cellular metabolism, glycolysis, Krebs cycle. We're oxidizing carbons or carbon-based molecules, right, to extract the energy. And then we're taking that energy and putting them into other molecules, most commonly NADH. But here on top of that, we're also creating a high energy intermediate. So that's kind of cool. So there's kind of like two like uses for the oxidation, right? To phosphorylate and to create that high energy electron carrier. Right? Why did we need to create such a high energy unstable intermediate? Well, we're going to use that energy to do something useful, right? In the following step. We are going to use the energy of this high energy intermediate, right, to do something very useful for us, right? And this here is where we have the first ATP that is produced in metabolism, right? We're going to take 1,3-biphosphoglycerate, right? We're going to take our enzyme phosphoglycerate kinase. For short, we call it PGK. Almost sounds like a, like a movie rating or of some kind. Right. Once again, with our divalent cation cofactor, in this case, magnesium, right? And we are going to create an ATP molecule, right? So we're essentially going from a very high energy, unstable molecule, and we're using that energy, transferring it to an ADP molecule, storing that energy in ATP in the end, and now we end up with a much stabler molecule, 3-phosphoglycerate, right? 3-phosphoglycerate is much more stable because it can be resonance stabilized. All right, so a lot going on here, right? But you see, like, things are making they make sense if we take the time to think about why each step happens, right? And it's okay if it doesn't make total sense for you at the moment. Just make sure you put a very strong emphasis on understanding the steps instead of blind memorization, right? That was a mistake that unfortunately I made when I first started learning glycolysis and the Krebs cycle. I was just trying to shove everything in my mind without any thinking, right? You've got to think big picture, and then you see as you start to like learn why things happen, right? We tend to remember it a lot better. So as a recap, in this step 7, ATP is generated, right? It is a substrate level phosphorylation, right? This is a fairly fancy thing that we're saying. The reason we're calling it phosphate level phosphorylation, substrate level phosphorylation, is because the vast majority of ATP in cellular metabolism is created using the proton gradient from the electron transport chain in oxidative phosphorylation using ATP synthase in the mitochondria. Right? Substrate level phosphorylation is where the enzyme is directly making our ATP, right? Instead of, using oxidative phosphorylation and the proton gradient. And of course, this is once again reversible because it's not one of our, 3 main irreversible enzymes. Now, of course, just because it is reversible doesn't necessarily mean that it will go backwards very easily, because as we talked about, this is a very exergonic process, right? Going from a high energy intermediate to a low energy resonance stabilized molecule, right? Which heavily favors the creation of 3-phosphoglycerate product, though technically it still is reversible. All right. So where do we stand? We've currently created an NADH molecule, and essentially remember we've created 2. Because we have, for 1 glucose, we have 2 glyceraldehyde 3-phosphate molecules. So we've created 2 of these. We've created 2 ATPs, and now we're on phosphoglycerate. That's 3-phosphoglycerate is where we are. All right, we're going to fast forward a little bit. Not too much interesting going on. We've got a mutase that's going to turn it into 2-phosphoglycerate. Then we're going to take an enolase and create a phosphoenol pyruvate. I find this step really interesting. Why, you may ask? Well, in step 10, right, we start with phosphoenol pyruvate. This molecule, right, contains the highest energy phosphate bond, right, in any molecule across any organism, right? So if we're just talking about humans, right, The highest energy phosphate bond that we will find in our entire body will be in the molecule phosphoenol pyruvate. That's really cool, right? Why is it cool? Well, we could use the energy stored in that bond, right, and transfer it, right, into usable energy. And that's exactly what's going to happen. You see that once again in this final step of glycolysis, we are going to be creating an ATP molecule, right? So the big enzyme here, pyruvate kinase, right? And you're starting to see the trend, right? These enzymes always have a little friend, a little divalent cation cofactor to help them out, right? Helps shield our reaction from our negative electron clouds, right? So pyruvate kinase, very important enzyme to remember because it is the third irreversible step that is catalyzed by this enzyme. Sorry, it's the third irreversible step, and this step is catalyzed by pyruvate kinase. Right, so we're going to be creating pyruvate, which is essentially, all right, the 3-carbon molecule that is the output of glycolysis. Irreversible, so now we can't go backwards. Can't take pyruvate, turn it into glucose directly. We're irreversible. So what we see is ATP is generated, right? We once again have substrate level phosphorylation, meaning that we're directly creating our ATP, right, where our energy is stored, right, instead of using oxidative phosphorylation in the proton gradient. And of course, irreversible. Cannot state how important this is. Super, super important. All right. So that's pretty much it for glycolysis. Let's finish off with a little talk about regulation of this molecule. Fructose 1,6-biphosphate, right? Which, if we toggle our memory, seems a little familiar. Yeah, we saw that earlier, right? We saw this earlier, this molecule, right? It actually has a positive stimulatory effect on pyruvate kinase. This is what we call feed-forward activation, right? This is where things that are back in the chain, all right, start yelling at things down the chain to get moving, right? Like this happened to me at the grocery store the other day. There was a huge lineup at the grocery store. I was second in line, right, just before the cash. There were about 10 people behind me. And for some reason, right, the person at the front of the line was like really not committing to going to a particular cash. And the line wasn't moving. It was getting backed up behind us. So, you know, there were empty caches and, you know, someone had went and cut from like a different angle, and the people behind me were getting mad. So they started yelling behind me at the person at the front. They were like, get her, move on, hurry up, let's go, get out of the way, move forward, right? And that's the same thing. Fructose 1,6-biphosphate is basically the people at the grocery store standing behind me being like, yo, let's go, right? We got some stuff building up here. Cool. So to summarize, one glucose molecule, right, we saw breaks into 2 glyceraldehyde 3-carbon molecules, right? So one glucose molecule will net 2 NADH molecules. So we put that here in the net. What's the net result from glycolysis? We got 2 NADH molecules, 2 ATPs. We produce 4 ATPs, but we had to use 2 at the start. So we produce 2 here, 2 here, right? 1 from each 3-carbon molecule. So 4 in total are produced, but we needed 1 here at the top for each of these steps. Cool. So we have a net 2 ATP that are produced. And we end up with 2 pyruvate molecules, right? Which will be used further down our plant. Right, so that's essentially glycolysis in a nutshell. Your big focus should be on the main enzymes. And just to make sure that we've kind of got this down pat, or at least starting to, you know, internalize it, let's look at a couple questions. All right, so everyone just take 30 seconds. I'll just chill here on camera to read this question and think what you would pick. All right, so this is a really nice MCAT question. All right, which of the following, all right, if we increase the concentration of, would have the least effect on the activity of phosphofructokinase? Now, if we remember phosphofructokinase, well, we had said it was the rate determining step, you know, and we saw that molecules associated with, low energy stores would stimulate it, and molecules associated with high energy stores would have an inhibitory effect. If we look at these, all right, we just say, okay, well, which of these is related? We're kind of looking something about the ADP ATP ratio. This is what we're talking about, ratio of high, like molecules associated with low energy stores versus high energy stores. Well, this is the only one that's not related, inorganic phosphate, right? AMP, ADP, ATP all have an effect on phosphofructokinase. Inorganic phosphate does not. It does not, right? Because we are not creating any ATP at this step. So that wouldn't have any effect. Right, remember, if you have any questions, right, please ask in the chat questions to Owen and questions to Carrie. Right, so if anyone here had picked C, right, ADP, remember ADP is important. It wasn't literally on the slide, right? It wasn't literally on the slide. We had AMP on our slide, we had ATP on our slide. ADP, right, is basically adenosine triphosphate, but that's been used up, right? Sometimes when you use it up, it comes up as AMP, so other times as ADP, depending on the circumstance. Right. And the second question, just to make sure, or not really make sure, but to help us remember and consolidate what we talked about, which of these enzymes catalyzes a reversible reaction in glycolysis? And remember, there were 3 we said were irreversible cornerstones, all right? See if you can spot the one that is reversible. All right, so which enzymes are irreversible? Well, we saw hexokinase, right? First one at the start that traps the glucose in the cell. We saw phosphofructokinase, the rate determining step. And we saw the final one, pyruvate kinase. We create that final ATP right there. Pyruvate kinase, the last step of glycolysis. So that just leaves the only one, phosphoglycerate kinase, right? So what this question nicely demonstrates is how sometimes on the MCAT, we don't look for the one the question's asking about, but sometimes we eliminate the 3 wrong answers, right, by using core knowledge. You see, there are too many enzymes to think about and learn them as reversible. Much, like pun intended, much easier to learn the reverse and learn which ones are irreversible. That's our goal. All right, so you guys are doing great. Honestly, if you got these questions wrong, doesn't mean you don't understand, right? If we're learning this for something that's way back, or if you're learning this for the first time, right? Remember, when you're doing questions in your practice, right, it's really the no judgment zone. We cannot measure our progress solely based on right and wrong in the short term. It's just a good way to, get discouraged for absolutely no reason, right? So someone asked in the chat, would you say these questions are as difficult as questions would be on the real MCAT, or is this simpler than a real question? Well, that question, to answer your question about questions, this question is definitely fair game for the MCAT, right? Now, the thing is that this question here doesn't require any passage analysis or reasoning. It's just straight up science knowledge. Right? So this is totally fair game for the MCAT. It's definitely representative of an MCAT question, right? But it's not representative of all MCAT questions, right? We can have things that are passage-based that'll be trickier, right? You may even have questions that are easier, right? So it's just to recap your question, it is representative of an MCAT question, just not all MCAT questions. Right. So now let's move on to learning goal number 2. And thank you so much for the questions coming in. Cool. Learning goal 2. In this section, we're going to talk about the citric acid cycle, right, and its regulation. Now, of course, before we can get to the citric acid cycle per se, right, we do need to talk about how we go from pyruvate, which was where we just left off our story, right, at the end of glycolysis, and how we get from that to acetyl-CoA, because that is the input to our Krebs cycle. All right, so why don't we get started? It all happens in a step called pyruvate decarboxylation, right? The name actually tells us exactly what happens. We go from our molecule pyruvate and we remove a carbon, right? So we're going from a 3-carbon pyruvate molecule to a 2-carbon acetyl-CoA molecule. We are effectively decarboxylating our pyruvate, right? So that's what's going on in our little picture here, right? A lot going on. So if you're studying this for the first time or even just studying this for the MCAT, remember to focus on big picture first, right? Trying to worry about the little cofactors associated with a pyruvate dehydrogenase complex, right, is a strategy to lose your mind, right? And think about it. If you were to spend 2 hours studying all of this really solid, right, in a week, you won't remember half of it, right? Not because you suck. That's just how human memory works, right? The key to human memory is repetition, right? So focus on the big steps. And then once those make sense, you'll notice it's a lot easier to attach knowledge once you have a solid base, right? So like trying to worry about lipoate or TPP here, right? Just not really the way to go. All right, so what's going on here? So like we mentioned, we're going from a 3-carbon molecule pyruvate to acetyl-CoA, which is 2 carbons. We're going to be, it occurs in the mitochondrial matrix, right? It's going to generate 1 ADH molecule per pyruvate, and therefore 2 NADH molecules per starting glucose molecule. And essentially, yeah, its raison d'etre is that it produces the principal substrate in the citric acid cycle. Now, unlike the enzymes we were looking at before, the enzyme here, pyruvate dehydrogenase complex, truly is a complex of enzymes, right? It's made up of 5 enzymes, right? And a multitude of cofactors. We've seen our friend magnesium before, right? TPP is a derivative of thiamine, which is vitamin B1. Vitamin B1. Right? It's kind of cool. Just little fun fact: there are diseases that cause significant issues, seeing as it hampers this whole metabolic process if we have vitamin B or thiamine deficiencies. Right? If you've heard of the condition called beriberi disease, it's linked to thiamine deficiencies. Yeah. So just a little fun fact. Side note, not important for the MCAT. So yeah, multitude of cofactors are important. This here is a complex made up of many enzymes, right? And you'll notice that we're going to be attaching coenzyme A. So when we say CoA, right, what we mean is coenzyme A. And what's significant about coenzyme A is that you see here we wrote it with the SH, right? It's got a thioester, right, in it. And this is important because thioesters contain really high energy bonds, right? And keep this in mind for the future, because we're going to come back to this thioester and its high energy bond. Remember, as we talked about earlier in glycolysis, when we have high energy bonds, right, we can use that energy, right, to do some cool stuff. All right, so the overall result here is pyruvate, right? Well, then we end up getting acetyl-CoA. Now, so yeah, we'll go from 3 carbons to 2 carbons. In terms of regulation, it seems like we're throwing a lot of information here on the board. However, many of these, all right, follow common sense, right? So this is why understanding right, is valued above all above memorization for the MCAT. So we see what type of things would have a positive stimulatory effect, right, on pyruvate decarboxylation. Well, this here is important when we're about to go into the Krebs cycle, make some energy. So it stands to reason when we're energy depleted, right, that we would want to get this going. So ADP, NAD plus, right, two molecules that are associated with a energy depleted status. And interestingly, calcium, right? Why do I say interestingly? Well, calcium is not necessarily associated with energy depletion. However, calcium is released by the sarcomere in the muscle when we're using that muscle. And you know, when you're running, right, or you're using your muscle, well, what do you need? Well, your body's like, yo, I need some more energy. So calcium, which is released with muscle contraction, actually stimulates the production of molecules that are going to end up giving us more energy. I find that one really cool. And consequently, NADH and ATP, right, the partners of NAD plus and ADP respectively, but they're high energy partners, will have an inhibitory effect on the pyruvate dehydrogenase complex. So you see, this is new information, right, but not a new concept. Fairly standard is that a product will have a negative feedback as well on the process that creates it, right? This is actually a little bit more interesting than just straight up, hey, I made a lot of acetyl-CoA, so I'm going to, turn off the system that makes more. And the reason is that acetyl-CoA here can actually come from different sources, right? It can come from most commonly fatty acid oxidation. So like say in the liver, you are breaking down fatty acids and you're creating a buildup of acetyl-CoA. It stands to reason that you would stop the other process that makes acetyl-CoA, right? So the negative feedback of acetyl-CoA on the PDC here is mainly utilized for when we have the production of acetyl-CoA through other sources, right? Mainly fatty acid oxidation, right? It can happen though through the breakdown of ketones and alcohols, right? Though those are not highly common situations. Right, so where do we stand? We have acetyl-CoA now, and now we're ready to roll because acetyl-CoA is our entry into the citric acid cycle, right? Also called, Krebs cycle, and also called the tricarboxylic acid cycle, right? One thing I did forget to mention, and I apologize, is that pyruvate breaking down acetyl-CoA is an irreversible process as shown by the unidirectional lines, right? And notice that we lose the carbon dioxide there, right? And that's why we went from a 3-carbon to a 2-carbon molecule. Okay, so we just did all this work. We started off with glucose at 6 carbons, and we broke it down into 2 3-carbon molecules, which then got broken down into 2 2-carbon molecules. What happens next? Well, we're going to undo all that work, right? Not for no reason. We're going to create citrate, right? So this is the first step of the citric acid cycle. We're going to take our acetyl-CoA, our 2-carbon molecule, and we're going to combine it with the molecule oxaloacetate in order to create citrate, right? Once again, right, this is an irreversible step, right? Notice the one-way arrow. The key enzyme here is citrate synthase. Right now, in terms of the cool facts that we try and learn about every little enzyme, originally when they started naming enzymes called synthase were enzymes that can catalyze the creation of new covalent bonds without using energy from nucleotides such as ATP. Right? So that's what's going on here. We're merging these two molecules together, right, without any extra energy, outside energy input, right? That naming convention no longer holds absolutely. They, the enzyme and chemistry people got together at some conference and they were like, all right, cool, you don't need to follow that anymore, right? We have a lot of exceptions. So overall, we're taking our acetyl-CoA, 2 carbons, merging it with oxaloacetate, a 4-carbon molecule, and we're creating a nice 6-carbon molecule, right? That's going to get oxidized as we go through the Krebs cycle. Cool. Right. Regulation is what we care about. So occurs in the mitochondrial matrix, condensation reaction, right? Notice here it says, sorry, condensation reaction. I just want to be clear. We're talking about the organic chemistry reaction in aldol condensation, right? Because as we know, condensation reactions don't have inputs of water, right? Condensation reaction is when you remove water from a molecule. Right here, it's an aldol condensation, right? Which is as you progress through your MCAT studying, you'll see that in your Kaplan organic chemistry books. And of course, as I mentioned, irreversible. In terms of regulation, right, see, new info, but not a new concept, right? When we understand, things start to repeat themselves. So the molecules associated with lower energy states, NAD plus ADP, they stimulate this. And the molecules associated with the higher energy states, ATP, NADH, once again, just like our previous steps, negative feedback, citrate, negative feedback on citrate synthesis. And here, this is the new one. This is maybe the one peculiarity of the step is that succinyl-CoA, it's an intermediate further down the Krebs cycle, has inhibitory effects as well on citrate synthesis. All right, so this is where we stand. All right, we now have citrate. And we're going to turn citrate into isocitrate, right? This is a reversible step. So we take citrate, right? We're going to be removing waters, adding waters, right? Through an intermediate cis-aconitate, create isocitrate, right? The key enzyme here is aconitase, right? Which is a ferroenzyme. It has an iron complex. Right, the exact reasons for the existence of this specific complex beyond the scope of the MCAT, but it is kind of cool if you want to look into it, just for a little side fun, right? What is the fun fact or cool fact that we are going to remember about this process? Well, it's going to be why we're doing this. Why are we changing citrate to isocitrate? Right, like almost every step in the Krebs cycle, it occurs in the mitochondrial matrix. It's an isomerization reaction. We're just changing it from one isomer to another. And that leads us to what that cool fact is. Notice that citrate, right, is a tertiary alcohol, right? Here's our alcohol, tertiary, right? And isocitrate is a secondary alcohol. So why did we just go through this whole process? It creates a molecule that is much easier to oxidize and therefore extract the energy. That's the point of this step. Right, and remember, focus on big picture, right? The MCAT's unlikely to ask you about why we did this. It's also very unlikely to ask you about cis-aconitate, right? They like the big steps, right? And this one here is not one of them. Speaking of big steps, well, that gives us a really nice lead-in to step 3 in the Krebs cycle, right? We're going to be turning our isocitrate molecule, and we are going to be oxidizing it into alpha-ketoglutarate, right? We're going to be extracting the energy by breaking those carbon bonds, and we're going to be creating carbon dioxide, and we're going to be storing some energy in NADH. So everyone take a minute to think about what's happening to the number of carbons in our starting molecule isocitrate. Remember, we're at 6 carbons. Where does that bring us now? Well, we're losing a carbon dioxide. That means we're down to 5 carbons. Cool, so the big enzyme here is isocitrate dehydrogenase, right? It seems like we're piling on the things that we need to learn, but remember, we are creating an NADH, and as we said before, in order to do that, we need to reduce an NAD plus molecule by dehydrogenated the isocitrate. So really this, if you know this, the input to this reaction, isocitrate, and you know that it's a step that creates NADH, then by default, we know the enzyme, isocitrate dehydrogenase. Right? Here the cofactor is manganese, right? But it could also work with magnesium in certain circumstances, right? It does happen through an oxalosuccinate intermediate, but big picture here, production of an NADH, right? And also production of CO2, which slashes the number of carbons. And just here, when it says first step to generate NADH and then first step CO2, they're not talking about steps inside this reaction. It's talking about it's the first step in the Krebs cycle to produce NADH and the first step in the Krebs cycle to produce CO2. Right? So this is one of the big steps. It's one of the really, really big steps. All right? Big steps. In fact, it is the rate determining step in the Krebs cycle. Right? So once again, we have our usual players stimulating the action, and we have our usual players inhibiting the action. Right? Hopefully you guys are starting to see some trends now. Great, so where do we stand? We have a 5-carbon alpha ketoglutarate molecule. Once again, just by looking at the picture here, just look at this picture. Try and anticipate what you think we're creating an NADH, right? What you think the name of this enzyme is going to be in our next step. This alpha KDC, what do you guys think that's going to be called? Think about it, and let's check it out. So what we're doing now, we're taking our alpha ketoglutarate, we're producing an NADH and losing a carbon. So we're oxidizing one of the carbon-carbon bonds, right? Taking that energy and storing it in NADH for later. And, well, look at that. It's called alpha ketoglutarate, our input dehydrogenase complex. So again, we have this dehydrogenase every time we're creating an NADH. All right, and here comes our CoA with our thioester bond. We're putting it into our molecule, and now we have a molecule that has our coenzyme A with that high energy sulfur bond. Interestingly, we will also note that some of the cofactors to this complex are the same ones we saw in pyruvate decarboxylation. Cool, so once again, right, like it's all been listed, but so far, everything in the citric acid cycle has occurred in the mitochondria, right? It is irreversible, that one's key, irreversible. MCAT loves the irreversible steps. And it's the second step in the Krebs cycle to generate NADH. So as well as the second step to generate carbon dioxide. So at this point here, this is the second step to generate carbon dioxide. That means from our starting citrate of 6 carbons, we're now down to 4. We now have a 4-carbon molecule. 4 carbons. All right, and once again, the usual suspects stimulate and the usual suspects, all right, have an inhibitory effect on this complex. We had mentioned before, right, if you remember in the first step when we created our citrate, I had mentioned that succinyl-CoA has an inhibitory effect. This is simply demonstrated here on the slide as for a second time. All right, so we're down here at the bottom. We have succinyl-CoA, and we're going to be creating succinate. Now remember, I had mentioned CoA has a thioester, very high energy, and how breaking that thioester, right, releases a tremendous amount of energy that can then be used to do some cool stuff, right? So this is what's going on here. So we're taking our succinyl-CoA, we're turning it into succinate, and we are going to be using the energy stored in that bond to do something cool. That something cool here is the production of GTP, right? GTP is it's a nucleoside triphosphate, right? High energy, very similar to ATP, right? And here we have succinyl-CoA synthetase is the enzyme that's making this happen, right? GTP from a metabolic standpoint is pretty much counted as an ATP because one thing that happens after this gets made, once again, substrate level phosphorylation, is it gets turned into ATP, right? There's another little enzyme called I think nucleoside diphosphate kinase, right? That converts the GTP that we make into ATP. So we throw it in to the pile of, energy that is extracted from our initial glucose molecule. Good stuff. Right, so big thing to remember here, we use the energy from the thioester bond and we then transfer that energy into a GTP molecule. All right. And that's what's there at the bottom. Right. Interestingly, it's the only step in which GTP is directly generated in all of glucose metabolism. Right. And it quickly gets turned into ATP, as we had mentioned. Right. So you see, like, notice what we're doing. We're putting an emphasis on trying to find cool little facts to remember every little step, right? And maybe you won't remember them tomorrow, right? But hopefully you remember a few. And then when you revisit them, it'll make it easier to remember and to assign to your long-term memory. So we're getting pretty close to the end point here of the Krebs cycle. We're over here in the bottom left at succinate, and now we're going to move on to fumarate. All right, so succinate going to fumarate. This is extra cool because it's the only step that doesn't occur, right, inside the mitochondria matrix itself, right? So you see, once again, we're producing a high energy electron carrier, right? In this case, it's FADH2 instead of NADH, right? So, all right, well, what's the enzyme's name? You see, well, we put in succinate, and boom, we're taking the hydrogen from it, so it stands to reason that the enzyme is called succinate dehydrogenase. All right. What is interesting about this? I said it doesn't happen inside the mitochondria itself, but rather on the inner mitochondrial membrane. Right. Succinate dehydrogenase is part of complex 2 in the electron transport chain. Right. It's actually bound to our FAD and our FADH2. So the electron carriers are actually part of the enzyme itself, which is kind of cool, right? And the fact that it's bound to an FAD means that this enzyme is classified as a flavoprotein, right? That's a term for enzymes that are bound to FAD, right? So this is kind of cool because the high energy electron carriers that are created in this process are directly delivered, right, to the electron transport chain, because the enzyme is part of one of the complexes in the electron transport chain. Anyways, if you guys end up, trusting Kaplan with your prep, we can learn more about this in the MCAT channel lesson on electron transport chain. Cool. So as I said, membrane protein, part of the electron transport chain itself, and FAD is covalently bound to our succinate dehydrogenase, which makes it a flavoprotein. And it's the only step that produces FADH2. Now, if you're wondering, why does it use FADH2? Why not use NAD+ and NADH? Like, basic, we already got that going, why change it up? Well, all of this, right, is governed by oxidation and reduction. And determining if something gets oxidized, reduced, right, is all based on relative strengths, like their reducing and their oxidizing power, right, which we measure with reduction and oxidation potentials. And the bottom line is that while we take some of the energy here out of succinate and use it to reduce FAD+, right, the reducing power is just not strong enough to reduce NAD+, right? It's just not strong enough. So instead of wasting that energy, we say, okay, it's not good enough for this, but it is good enough for that. It's kind of like, man, I just don't have enough money to sit in first class. Well, I don't want to not go on a trip. I'll just go sit in coach, right? It's kind of what's going on here. All right. So now we're up to fumarate, right? So we got fumarate. Notice at this point here, we've created an NADH, another NADH. We've made a GTP, which gets turned into ATP. Now we have our FDH2. We're almost done. We're at fumarate. So fumarate gets turned into malate, right, by the enzyme fumarase. This is the least interesting, in my opinion, right? Don't go telling your scientist buddy who does his research on this. I'm not standing by my claim. I'm just saying this is the least interesting step. Nothing really cool happens, right? We're just adding a water molecule. Now, we said we needed something cool to remember every step. So why don't we put in a side story, right? There is a disease, right, that is caused by a defective fumarase enzyme. Right? It's an extremely rare autosomal recessive disease, right? It's fumarase deficiency. Up until the 1990s, there were, I think, roughly like 10 recorded cases of it, like super, super rare. And then they found a massive cluster of individuals that had fumarase enzyme problems, right? And the reason they found 20, right? 20 people that had this. And this was in a community in Utah and Arizona, right? They were part of the Church of Latter-day Saints, right? Because those communities tend to, reproduce in isolation. They were basically in reproductive isolation, and therefore had, I don't really like to use the term inbreeding, but they're just lack of genetic diversity, shall we say. And a lot of, unique diseases and rare diseases tend to come up in communities that display reproductive isolation, such as, Mormons in Utah. There's certain ones with in Amish communities as well, and as well as like where I'm from in Quebec, and especially in some of the more like rural communities. Definitely a lot of cases of unique diseases. They actually come up in like medical textbooks. There was, or when they do, a couple of my friends were in medical school, and they say, oh, they keep bringing up Quebec in some of the exam questions, because it's fairly common due to the reproductive isolation. So anyways, no real cool story about this step, but we just threw in a little side note so we can try and remember. So again, once again occurs in the mitochondrial matrix, just like every step except the last one we just saw. We add an H2O. And that finally brings us to our last step. We're just going to simply reform our oxaloacetate. So we're reforming our oxaloacetate, malate, all right? Oh, what's going to happen? What do we think this is going to be called if we're making an NADH? Everyone take a moment to try and think that through. Yeah, it's called malate dehydrogenase. So we're taking our malate, we're dehydrogenating it, adding that hydrogen to the NADH into a high energy electron carrier, right? And now we've reformed our oxaloacetate. We're ready to reform the cycle. This is the final step. Sorry, just to go back here. One thing that is kind of cool is that while we are now back down to a 4-carbon molecule, right? The carbons from the acetyl-CoA that we initially put into our cycle and combine with the oxaloacetate, right? The 2 original carbons that we merged with the oxaloacetate are now actually contained in our remaining oxaloacetate here. So the carbons that we lost were not the ones that we put in, right? And they know this by having done experiments using like radioactive labels on the carbons, or radioactive isotopes, right? So it's kind of cool. Don't need to know that for the MCAT, but just a fun fact. So the carbons that we put in are still there, and they're going to disappear in future steps. Cool. So overall, in summary, right, we talked about how when we have adequate energy, right, and that is signaled by molecules such as ATP or high energy electron carriers, NADH, that's going to have plenty of inhibitory effects on many steps. You will notice that all of these inhibitions, whether it's on our pyruvate decarboxylation, or citrate synthase, or isocitrate dehydrogenase, or alpha ketoglutarate dehydrogenase complex, the negative inhibition occurs at the level of all of the irreversible steps. Notice that it's only the places with one-way arrows. That can help us with our memorization. And our energy deficient molecules, or molecules indicative of such a state, right, they're going to have an inhibitory effect, once again, at all the same places. All right, so this is all pretty cool stuff. Right, we talked about acetyl-CoA having a negative inhibitory effect on PDC. We talked about that. Citrate, negative impact on its own enzyme, right? And this year we had talked about succinyl-CoA inhibiting the previous molecule that makes it, but also, right, has a inhibitory effect on citrate synthase. All right, so this is just some summary, some recaps, and so on. Right, we talked about the effect of calcium, right, because you release calcium when you contract muscles, right? So that's going to have inhibitory effects at a few places. Kind of cool. Notice once again, mainly the irreversible steps, right? The rate determining step here, and the next one where we're making the carbon dioxides. So yeah, I find that helpful to remember for when the calcium acts, right? Notice the calcium is acting at the 3 places where carbon dioxide is produced. So yeah, in summary, in terms of energy, here are all the things that we're making. We're producing 2 NADHs per glucose molecule in pyruvate decarboxylation. And then we're making 6 NADHs per glucose molecule, 2 FAD pluses, FADH2s, and 2 GTPs. Right, so if you wanted to calculate the net, it's all here. For every 2 pyruvate used, we make 2 NADHs, 2 acetyl-CoAs that are getting made in our pyruvate decarboxylation. And then in the Krebs cycle, for every 2 acetyl-CoA used, which comes from 1 glucose, 6 NADH, 2 FADH2s, and 2 GTPs. Quick note on FADH2, it creates less net ATPs in electron transport chain than NADH because it enters the electron transport at a later stage. So that brings us all to this cool question. All right, everyone take a moment to think this one through. Right, so this is typical of an MCAT question, right? Because they're trying to throw us off with like plants, and you're like, man, I didn't study plants. Oh crap, glyoxylate cycle, I didn't study that. Oh my God, I'm gonna fail, my life's over, right? Like it's very easy to have such thoughts, right? One thing I like to teach always my students is that sort through this nonsense and really figure out what the question is really asking. This question is asking, if there was a problem with aconitase, well, what would happen? What would we change? So really this question is just we need to think, what reaction does aconitase catalyze? Turns citrate into isocitrate, boom. All right, let's take a look at this next question here. Take a moment to read it, you don't need to answer it. All right, I just wanna show you something cool. Right, so you see here, they're throwing in a hypothetical. MCAT loves hypotheticals. Now what we would have to do? Work the Krebs cycle backwards? My goodness, right? So like if you really wanted to, like say you're elite, right? You do this, okay. What are the steps going from succinate to oxaloacetate? Well, you go from, succinate, FAD, blah, 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 okay, right? Like all from memory, right? And you get to C as the answer. Fine, right? I'm going to show you type of MCAT reasoning that shows that you don't necessarily always need to master all the details, right? One thing that we should know is that the creation of ATP, right, occurs when we go from succinyl-CoA to succinate. That's a major step in the Krebs cycle, right? So we should be fairly comfortable eliminating A and B and saying that there's no ATP involved here. Right, and in fact, when I say creation of ATP, it's really GTP and then ATP indirectly. So we eliminate A and B. We eliminate the low-hanging fruit. And then instead of trying to wrap our head around when does the NAD plus or the NADH occur, we can approach this conceptually, right? Meaning, let's look at this molecule, oxaloacetate, and let's compare it to succinate. We're using big picture concepts. When you add bonds to oxygen, you oxidize. When you remove bond stop from oxygen, it's a reduction. So you see here, we have an O double bond to the C, which is missing here, which means we're losing bonds to oxygen, which means we are reducing this molecule, right? We produce NADH when we oxidize molecules. So since we are reducing this molecule, that means we are not producing NADH, and that's why we pick the other answer, production of NAD+. Right? Don't worry if I found that tricky, right? Just trying to emphasize and illustrate with this very difficult problem, right? In case anyone's asking, like, how does this compare to the MCAT? This is like as hard as it gets, right? But this very difficult problem can be solved without complete total mastery of all the little details in the Krebs cycle, just like big picture concepts. All right, so that brings us till the end of our lesson. Here's a little overview. I did lots of overviews. Sorry, I don't want to keep you guys too late. So just a quick note as we conclude the end of this lesson, just give you guys a few tips for success. Follow a study plan and don't just do blind studying, right? You want to build up a study plan that has a huge emphasis on full-length practice tests, right? Sources of expertise, right? And mix content and practice. This is really, really important. Keep it fresh, right? If you're looking where to, get all this practice and solid material, Kaplan has a great program, and I've been with Kaplan for a long time, right? So I really trust the program. Seen lots and lots of success, right? Kaplan has over 2,000 MCAT questions just in the adaptive quiz bank that tailors to your particular level, right? It gives you access to all of the full-length tests that they have, plus all of the AMC materials. They're included as part of all the Kaplan courses. That's huge. All the AMC tests and their practice tests, right? Kaplan has science review videos. It has dozens and dozens of lessons, just like the one you just had, on all the topics, right? And they also can help you with your medical school admission, helps with your statements, your interviews, and so on. Cool. So what's important is that you choose the instructions that's right for you, right? Whether you need class with a live instructor like myself, you want to join one of my classes, or you just want the Kaplan materials, or you'd like a coach or a tutor, we have things that tailor to everything, right? You can go on the Netflix, is what I call Netflix for the MCAT. You have an MCAT channel every day. Well, live lessons 6 days a week, but you can watch it 24/7, right? Lessons just like today. And a lot of our students just love the MCAT channel. It's really, really good. Over 90 lessons. So yeah, like I was saying, 16 practice tests, right? All the AMC practice tests, some good stuff. So I invite you guys to, join our classes, right? Or at least check out all the resources that are available. And apart from that, I don't want to keep you guys too late. So thanks so much for coming. If you guys have any questions at all, please don't hesitate to ask us in the chat. So on that note, thank you so much for coming, everybody. Have a great night and best of luck